Hello, this is Eva Amson, welcoming you to this Bite-Size Bio webinar, which today is sponsored by Merck KGaA Darmstadt, Germany. Millipore Sigma is the US and Canada life science business of Merck KGaA Darmstadt, Germany. Today's presentation is titled Unleash New Biological Insights with Innovative Glycoanalytics, and it's being presented by Morten Thijssen Andersen from Macquarie University and Mark Pavlicki from Millipore Sigma. Morten is an associate professor at Macquarie University, Sydney, Australia, where he heads the analytical glycoimmunology group. He is also a visiting professor at Nagoya University in Japan, where he leads the clinical glycoproteomics lab within the Institute for Glycocore Research. His research explores the roles of glycosylation in innate immunity and related diseases such as cancer or infection, using sophisticated mass spectrometry approaches. Mark is a senior product manager within the molecular reagents and tools franchise at Millipore Sigma, with focused portfolios containing biochemical tools including enzymes, proteins, and reagents useful in the fields of glycobiology, proteomics, biochemistry, and more. Mark has a background in glycobiology since graduate school, where he studied the functional impact of glycosylation on protein-protein interaction. As always, we will have a question and answer session after the presentation. Morten will not be with us for the Q&A, but if you please type any questions you have into the questions box at the top right panel of your screen, I'll put them to Mark at the end. So now over to you, Mark, for the presentation.
Thank you for that introduction, and welcome everyone to our webinar on cracking the sugar code and unlocking glycobiology insights through glycoanalytics. Before we get started on the core part of the webinar with Dr. Thijssen Andersen, I wanted to give a brief overview and introduction of the field of glycobiology. This is very brief. I won't do the full field justice, but I will do my best. And then after that, Dr. Thijssen Andersen is going to go into real-life application of analytical sciences toward unlocking insights into glycobiology. So let's start with a definition, because what falls under glycobiology can often be misunderstood, especially by people new to the field. And I wanted to paint a picture of where the guidelines are and the guide rails for what glycobiology is. So typically, research pertaining to carbohydrates and as a source of energy in our catabolism or the catabolism of organisms is not considered glycobiology. So glycolysis and things like that are not considered technically glycobiology. Whereas study of the oligosaccharides and their impact on structure and function in immunology, cell adhesion, enzyme activity, those are some examples of areas where it would fall under glycobiology. So it is the study of oligosaccharides and their function in biology, excluding carbohydrates for energy use. And then there's a gray area in between where we start to talk about things like glycation and in diseases like diabetes. So much of glycobiology research is focused on the immune system. Glycoconjugates, they play a central role in things like cell surface recognition, blood typing, immunogenicity, antibody interactions. And I want to point out there are three major classes of glycoconjugates, and within these, things can get more divided and complicated. But the three major classes are N-linked, O-linked, and lipid-linked. You see some examples on the top right there, and we'll go into a little bit on N-linked, and you'll see Dr. Thijssen Andersen go more detailed into the glycomics. And these play an especially important role in eukaryotes. N-linked glycosylation is very absent, or is nearly absent in prokaryotic organisms, but there are known cases. And then O-linked glycosylation and lipid-linked can be seen throughout kingdoms. Very important macromolecular structures. So what I've provided here are two very well-known examples of glycosylation directly impacting function of enzymes and proteins. So on the top here, we have RNase. This is a classic example where we even name the enzyme based on whether it is glycosylated. So RNase A is a non-glycosylated form, you see on the right there, and RNase B is the glycosylated form on the left. And research shows that as you reduce the size of the glycan attached to the enzyme, activity increases. And a fully glycosylated form of RNase is actually inactive. This is a model demonstration of the impact glycosylation can have on enzymatic activity. And then below, we have a classic case of immunoglobulin G and how glycosylation impacts it. So antibodies are extremely versatile molecules, and something that often gets forgotten when you talk about monoclonal antibodies and ADCs, which are common therapeutic modalities nowadays, is that they're all glycoproteins. So occupation of the CH2 glycosylation site in the FC domain is essential for FC receptor binding, for example. And many of the immune responses associated with antibody function rely on this interaction. So, and then even the type of glycoform is important as well. And so you see here in this example, for those of you that aren't used to looking at these shapes for glycans, that red triangle that's highlighted on the left is a alpha-1,6 linked fucose, a core fucose on that N-linked glycan. And its presence makes binding to the FC receptor very poor. And then its absence will increase the binding affinity 20 to 30 times. And that little monosaccharide, that little fucose, can make all the difference on whether an antibody elicits ADCC response. And that's really important to know when you're talking about immunology of antibodies. So just some examples. These are very well known and characterized examples where glycosylation has a huge impact on biochemistry and physiology. So. And now I wanted to give you an idea of why glycobiology is so difficult to study. What's the big deal? So the oligosaccharides, they may be unlike many of the macromolecular molecules we're used to studying. So we're all familiar with the central dogma of molecular biology, where DNA will be the template for RNA, RNA is the template for protein. And much of the phenotypic characteristics we see are a result of regulation of transcription or translation along that sequence of events in the central dogma. All of these are linear templated polymers. So that lends themselves to being able to be manipulated and altered for experimentation. So there's a way to do that. We have enzymes, we have tools, we have chemicals to do this. We can synthesize linear polymers of nucleic acids relatively easily. And whereas glycosylation is a post or a co-translational modification. It's not templated, and many times it's branching, and it has regio and stereo-specific moieties on the oligosaccharides and on the monosaccharides that are polymerizing. So glycosyl transferases and glycosidases and lipases all work in concert to produce heterogeneous mixtures of glycoforms. So we're talking enzyme activities and their levels in the cell and different organelles that all kind of come together to form all the glycoforms that we see. So any one gene that expresses a glycoprotein could result in a distribution of many glycoforms attached to the same conjugate. And this is called microheterogeneity. And in my cartoon here, you see how you can end up with multiple forms. And all of these forms are branching They're not templated, they're microheterogeneous, and they're polymers. Makes it very difficult to make samples, to run experiments, a difficult area to study. So we need tools to study glycobiology. And our goal is really to build tools for scientists in general and to find areas where tools are lacking. And glycobiology is one of those. And so I like to think about the tools used to study glycobiology in 4 categories. And you see them listed here: capture, release, modification, and analysis. And so highlighted, and so if you look at like capture, this would be things like resins and lectins, derivatized lectins that might recognize and have an affinity towards oligosaccharides used for pull downs or purification. Release category would be tools to separate the glycoforms from the conjugate, whether it's a protein or a lipid or RNA. So release tools. Modification would be things that are used to alter glycoforms. So this could be exoglycosidases, glycosyltransferases, maybe labels and things like that. So that's useful, especially when you're running experiments and say you want to remove all sialation from your glycoforms. There's enzymes for that. And then analysis is anything used on the back end for quantitation or qualitation. So to identify your glycoforms, glycoprofiling, and you'll see a lot of that in the talk. So and then I highlighted a few things, new products that or coming products that are in each of these categories, showing that we're trying to always add tools into these categories. You see the Suppel Carbon product is a chromatography tool for analysis of glycoforms that you're going to hear more about in the talk. Beta-galactosyltransferase is a modifying enzyme will add galactose to certain oligosaccharides. Mucinase is a neat product that is brand new. It will digest mucins, which are notoriously difficult to access large O glycoproteins. So it's a protease that is selective towards mucins. And then we have coming soon some recombinant kidney bean lectin, which could be used for oligosaccharide capture or recognition. So if you'd like to learn more, I would direct you towards our glycobiology, glycoprofiling category page. That QR code will bring you there. So will that URL there. There are a lot of products and guides there. There's sections that show you how to release N-link glycans and O-link glycans and various sorts of app notes and things. So it's a useful resource and it's always being updated. So check again in the future. And thank you for your time. And now to the core of the talk.
Thank you, Mark. And we will now hear from Morten.
Hello, I'm Morten Andersen, and welcome to this glycobiology-focused webinar. First of all, let me thank Corey Morocco and Michael Yee for the kind invitation to deliver this webinar and also to discuss some data surrounding our collaboration, where we're looking at how to use innovative glycoanalytics to expand our insight into the human glycobiology, mostly in this webinar focusing on glycomics-based explorations. First, let me take a minute to introduce my research teams and focus areas. So I'm heading the analytical glycoimmunology group based here at Macquarie University in beautiful Sydney, Australia. And as of recent, also heading the clinical glycoproteins lab at the IG core at Nagoya University in Japan. For a number of years, we've been interested in understanding the role of protein glycosylation in innate immunity. So we're interested in fundamental processes related to infection, inflammation, but also diseases such as cancer and sepsis, where the innate immunity is playing a significant role. Our approaches to studying this topic takes different directions. We can start with a more open-ended agnostic approach, looking at the diversity of the glycoprotein, using glycomics, glycoproteomics, and other types of omics methods. But we also often like to understand the glycosylation machinery. There's a complex web of enzymes that produce glycosylation, and here we're using other types of omics. We're often then interested in drilling into the biology of individual glycoproteins of particular interest, and here we can understand or try to understand the structure-function relationships using a whole host of other tools and disciplines. Rather than providing a long introduction to the field of glycobiology, I thought I would just mention this fantastic resource that we now have available, the Essential Glycobiology. This most recent edition of this textbook has become available earlier this year and is now freely available on this NIH bookshelf. And what it has done is providing a terrific resource into describing the consensus of how we're depicting glycans. We're using these symbols, the colorful symbols, as a way to quickly draw structures in the field, and that's really propelled the field forward. And I thought I would just briefly mention this particular chapter 51 that is of importance to this webinar today. So why study glycobiology? Well, all cells in our body are covered in a highly heterogeneous glycome. That's what we know as a glycocalyx. And what these glycans in the surface of cells are doing is that they facilitate communication to the environment between the cells, but also into the extracellular environment. That glycocalyx can be depicted as this more simplistic form here. And what you can see is that we have different types of glycoconjugates, glycoproteins, glycolipids, proteoglycans, etc. But what I'll be focusing on in this webinar are two types of glycoproteins: the N-linked glycans, where the glycans link through to the polypeptide chain through the asparagine residue, and the O-linked glycans, where the glycans link through a serine or threonine residue. And much of our work in our group is surrounding the characterization of these structures. And we need to be able to characterize glycoproteins as a prerequisite to understanding their function. But there's a lot of analytical challenges relating to glycoprotein characterization, as I've tried to indicate in this slide here. So looking at a relatively simple glycoprotein with two glycosylation sites, an O-linked glycan and an N-linked glycan. From an analytical point of view, there's many levels of insight we need to obtain. We need to understand what are the protein carriers of the glycosylation. So we need to identify the protein, we need to identify the site of glycosylation, both the O-linked site and the N-linked site. We need to understand the monosaccharide building blocks that are forming these N and O-linked glycans. We need to be able to understand how the building blocks are linked, so the topology and the linkage type. We need to understand how much of the glycosylation site is occupied, as well as the heterogeneity that can exist within a site. So all in all, a very difficult task, but we can now use glycoproteomics to understand many of these levels here. So glycoproteomics is defined as the study of the intact glycopeptides or glycoproteins, but there's some levels of structural insight that we can't get glycoproteomics, and here we have to resort to glycomics, the study of released glycans. And we're often using these two omics disciplines in tandem because they complement each other nicely. For glycomics, of course, we're looking at released glycans. So here we can use PNDSF as our favorite enzyme to release all of the N-glycans. And for glycans, we don't have an enzyme for that, but we can resort to chemistry to detach the O-linked glycans. So as you can imagine, from a glycoprotein mixture, we can then decide to take different routes. We can decide to take a glycan-focused route where we look at the released glycans using glycomics, and that's what I'll be focusing on today. We can also look at the previous glycosylated sites to simply identify the glycosylation sites, or we can leave the glycans on the peptides and then study them as an intact molecule to get more site-specific distribution information. Focusing on glycomics, what we're doing at Macquarie and have been doing over now more than 10 years is to focus on a so-called PGC LCMS method. PGC short for porous graphite carbon. So just in short, and I'll get back to this workflow later, starting from a glycoprotein or glycoprotein mixture, we can immobilize these proteins on a PVF membrane. Then we can detach the glycans that we're interested in. If it's in glycans, we would use the enzymatic approach. And then we basically leave the glycans underivatized with the exception of a quick reduction step where we reduce the reducing it of the glycans before a desalting on using SP type approaches and then down to the analytical step here, the PGC LC-MS profiling. And then we can use a number of different tools to identify and quantify the glycans before the more statistical and data visualization tools. So we're using mass spec in tandem with the LC component to characterize. Of course, using the mass spec alone has limitations given that we know glycans can appear in different isomers. They can be perfectly isobaric, as those two glycans indicated here, having exactly the same mass, same elemental composition, only differing in the sialic acid linkages here. We can then begin to fragment the glycan to get more sequence information. But for those two isobaric glycans here, that's still not enough. We'll get exactly the same B and Y ion fragments from these two structures. So in answer to separate those two type of isomers, we are then using porous graphitic carbon or PGC. Simply because PGC has a unique ability to separate glycan isomers by shape, as well as charge and hydrophobicity. In fact, it's a mixed mode separation, and we still don't really fully understand the separation mechanisms of PGC. But what we do know is it's separating based on these planar carbon inert sheets. And that's really how we obtain this beautiful separation of the glycans and glycan isomers as indicated here. To explain a little bit more about our characterization of the individual glycan structures, we're using three features. We're using the PGC retention time, as well as the molecular mass and the way they fragment in the mass spectrometer. So let's take an example, glycan A and B here, isobaric glycans, glycan isomers, exactly the same molecular mass, exactly the same elemental composition. The only difference is being the GLUCNAC being bisected here, and here's a triantennary structure. And you see how nicely separated they are on the PGC face. The tandem MS spectra do look a bit different, but not enough to tell them fully apart. It's really the combination of these three features that gives us that information. And over the years, we've worked out many different retention time rules for PGC that we are applying on an everyday basis. An example here from bovine fetwin, where we're separating isobaric end glycans all capped with sialic acid. So if you look at this panel here, the biantennary diisoylated glycans, and I hope you can appreciate how well we can separate the alpha-2 6-linked biantennary glycan from the alpha-2 3-linked biantennary glycans. Exactly the same molecular mass, but separated with well over 10 minutes here. But there are some shortcomings with PGC-based glycomics that we identified over the years, and that's that short glycans are often overlooked with traditional PGC-based approaches. We came to realize that in this study here in biomolecules in 2015, so a fair few years ago, where we did a profiling, a structural analysis of cathepsin G here that has this single N-glycan decorating its surface. So with a PGC-based glycomics profile, we observe these types of glycans, so both complex type glycan, but also short mannose type glycans, and even the fairly short chitobias core type as well. But then doing a profile of intact cathepsin G that is arguably more an accurate way of getting glycoprotein information, we saw this very high abundance of the ultra short glycans, both the GLUCNAC and GLUCNAC fucose that decorates this profile. Things that were totally overlooked with the PGC approach. Really meaning that we have overlooked these super short structures with PGC. And that's because PGC is not known to retain well or at all monosaccharides and disaccharides. In attempts to improve that, we've been working with a product they have on the market, supercarbon, which is a new type of PGC material that differs from the existing one by having a more narrow particle size distribution, but retaining some of the more classical features, such as the wide pH range and pressure profile. And what one of my postdoc has been working on over the years and month is to look at the separation capacity of these new supercarbon phases. So here, first we're looking at an O-glycan mixture from bovine salivary mucin. So this is a fairly well characterized mixture. We can see really neat retention and separation of even monosaccharide and disaccharides from this mixture, things that we wouldn't have seen with more traditional PGC methods, but still retaining this really nice separation of the salicylic acid glycoforms of the isomeric isomers. So really expanding the glycomics insight relative to more traditional PGC that we've been working with over the years. So based on that observation, we decided to really explore that retention ability of monosaccharide and disaccharides a bit further. So here Andrew's been looking at isomeric monosaccharides, so glucose, mannose, and galactose. These are reduced monosaccharides, and I hope you can appreciate the really nice separation between galactose and mannose, and even a subtle separation between glucose and mannose. We saw the same trend with reduced isomeric disaccharides. So here we were looking at lactose, maltose, and sucrose, showing a really strong separation between sucrose and maltose, and even subtle separation of the lactose and reduced maltose. We saw that the new phases, PGC phases, were reproducible in terms of their retention of the monosaccharides. Here we have a triplicate injection of non-reduced fructose showing near identical retention times for these 3 injections. We also saw, and here focusing on non-reduced monosaccharides, glucose and fructose, really well separation between these 2 isomeric monosaccharides, and also this clear separation of a reduced and a non-reduced maltose. And this is one of the latest data that we have obtained on these new phases. This is from Sponsal, a PhD student in my group, that has achieved a really nice separation between GLUCNAC and GalNAc, which is often a analytical challenge in our field. And having now an ability to separate and quantify mixtures of these 2 species will allow us to address sort of a range of other biological questions that we haven't been able to address previously. Much of this monosaccharide analysis has traditionally been made on high-performance anion exchange with pulse and perimetric detection, and having a PGC platform will greatly advance the field of monosaccharide analysis, and now also expand it to disaccharides and trisaccharides. So in short, it's a Pelcarbon PGC expands our view on the human glycome. So here's shifting gears to more application of how we're using glycomics to explore disease, and focusing on sepsis and septic shock because it's a focus in my team. Briefly, a way of introduction, it's an infectious disease, can be infected by any types of microbe, viral, bacterial, and fungal, mostly to the bloodstream, but it could be anywhere in the body. Sepsis is defined as a life-threatening organ dysfunction caused by a dysregulated host response to infection with a fairly high mortality. And it becomes particular a problem if the sepsis escalates to septic shock, which is a subset of sepsis with a greater mortality, as you can see here. Septic shock, unfortunately, has a very high prevalence. The incidence rate is even rising in the Western and also in the developing world. And it's a costly disease, as you can see here. The clinicians can't do much, and there's basically been no therapeutic advances for 30 years. So it's really important that we get a better understanding of the disease mechanism of the disease. So in one step towards understanding sepsis is to understand the precursor disease, which is called bacteremia. And in works we did a few years back now here, this was work performed by Dr. Sansini Chatterjee, a former PhD student in my group. We looked at bacteremia because it is a precursor disease to sepsis. Bacteremia is the presence of viable pathogenic bacteria in the normally sterile blood, and it can arise both from primary and secondary blood infections. Both gram-negative and gram-positive bacteria can cause bacteremia. Unfortunately, it's a fairly slow identification, slow diagnosis. Clinicians have at their disposal, involving blood cultures, which is tedious and lacks speed and sensitivity. So, and therefore, these types of illnesses can often escalate to sepsis and septic shock. So, having a way to quickly identify bacteremia is of interest to clinicians. So here we ask the question, can quantitative glycomics be used to stratify bacteremic patients from healthy donors? For that purpose, we had a number of individuals with bacteremia. This was work done with Professor David Tolpe at the Royal Adelaide Hospital. We had bacteremic individuals infected with different pathogens, both gram-negative and gram-positive bacteria, as well as a healthy control group. Looking at the PIT severity score, this is a score that indicates the severity of illness. You can see that these are not severely ill. The value here is well below 4, which indicates critical illness, and the scale goes up to 14. So these are only mildly sick individuals. We obtained blood and serum from these individuals, and then could do glycomics-based profiling of these serum samples using the method I already outlined, depositing the samples on PVDF membranes, producing the glycans after their release using PNGSF, and then profiling using PGC, LSMS in negative polarity mode. Getting a quantitative picture of the glycan isomers and their structures, from which we could use a more advanced statistics. This is the type of data we're getting from glycomics. So we're getting a glycomics map of the glycan fine structures that we find here in serum, mostly complex type glycans, but we do also see oligomonos and hybrid type glycans. PGC also gives us a quantitative picture of each of these glycans, and breaking it down to the highest level, so the glycan type distribution, you can basically see no real differences between the healthy group and the bacterial infection groups. We really need to drill down to the glycan fine structures, and this is where PGC becomes important because it gives us that information, and here we can begin to see stark differences in the total level of silylation, glycosylation, and branching. trying to use this data in an unsupervised way to see if it stratifies the healthy individuals from the bacteria-infected individuals. We can use hierarchical clustering. And here we can nicely see how the total serum glycone nicely separates the healthy individuals from the Pseudomonas infected individual that separates over here from the Staph aureus and Strep viridans infected individuals up there. So that was using the entire glycone, but we also saw that individual glycans can in fact differentiate between the pathogen types. Here we see glycan 9 stratifies nicely healthy individuals from E. coli. Glycan 11 stratifies healthy individual from aeruginosa. 12A, so that's the isomer, stratifies Staph aureus, as well as glycan 48 here, Strep viridans. But we really wanted to understand how the glycans, if they could stratify between the different pathogens at play here. So here we're looking at the glycans that are changing across the 4 different types, these 7 glycan species, and see a nice separation here of Pseudomonas aeruginosa infected individuals that are clearly quite enriched in glycosylation and depleted in sialylation. So also on a PCA here, you can see nice stratification of the Pseudomonas aeruginosa infected individuals. And here, breaking them down into the individual features, elevated glycosylation and bisecting glycoanalytic levels and depleted sialylation level relative to the other infection types. And in conclusion, we can see that quantitative serum glycomics accurately stratifies the bacterial patients according to their pathogen-specific features. So a question that Kevin and Liam has been taking on since is whether serum glycomics can inform on septic shock survival. So as I mentioned, septic shock follows an exacerbated post-response organ failure that can lead either to death or to recovery, and trying to make use of the glycomics to see if we can inform on the outcome here would be an interesting thing to do. So for that purpose, again, we teamed up with Torpy's team here at Royal Adelaide Hospital, got access to quite a number of septic shock survivor and septic shock non-survivor samples that we could follow in the ICU day by day until recovery or death, and then used glycomics, quantitative glycomics, to look at this progression. Again, we can obtain this glycome map from the septic shock serum. Here I'm just highlighting the most abundant, most prominent glycans structures. Because we have longitudinal information here, we can follow the evolution of the glycome of the non-survivors towards death or towards recovery for the septic shock survivors. We could basically see that in an unsupervised PCA that there's no real stratification between the survivors and non-survivors based on the serum glycome, which was a bit of a disappointment when we looked at it like this. Little bit of separation using unsupervised clustering, so the non-supervised non-survivors falling over here, but there was really no clear separation between the groups. Then, when we didn't give up, we used a machine learning approach where we used random forest in collaboration with Dr. Matt Trainey, where we used a training model to and random forest to train on all the ICU data points except for data point 1. And then we used this trained model to test on all the ICU day 1 data points. And using this model, we could see very high accuracy, a very high specificity. Basically 35 out of 37 patients were correctly classified using this machine learning approach and with a decent sensitivity. 6 out of 8 non-survivors were correctly identified. So just highlighting, wrapping up here. So I hope I've been able to show you that we believe that PGC-based glycomics provide unique insights into human glycobiology and the complex glycome. We've seen how this new carbon phase available can expand our view of the NNO glycome. It's especially these very short monoditriglycerides, is able to retain and separate. We've seen an example of how the serum glycome stratifies patient with bacteremia, and as a more recent work that's still unpublished, we've been looking at how the glycome differs in septic shock survivors and non-survivors, of course, opening up for new avenues for prognosis and clinical decision making. So with that, I'll thank my two research teams here, in particular Indris Komarami, as well as Nas Bansal, for their collaboration, Corey and Michael here, as well as Jeffrey for their collaboration, funding bodies, and thank you all for your attention, and I'm sorry I couldn't be with you here in person today.
Thank you, Morten and Mark. That was an excellent presentation. We have a few questions from the audience. If anyone else has a question, please feel free to post this in the questions box on the top right of your screen. As I mentioned earlier, Morten can't be here for the Q&A, but we'll send him any questions that are specific to his presentation. And meanwhile, Mark is here to answer some questions. So the first question I have for you, can't I just use PNGF to release all my glycans from glycoproteins?
Thank you, Ava. Yeah, that's a good question. And I think the short answer is, for N-linked glycans, mostly yes, but be careful, because while PNGSF is pretty versatile in its substrate specificity, it's very broad. There are limitations, and so if you don't know what your target is, if you don't know the structure of your glycan, you may miss out on things. And so for example, if there are core alpha-1,3 linked fucose, PNGSF won't work on that. If it's not attached to at least a tripeptide, the glycan, it won't work on that. And then the biggest hurdle, I think, with PNGSF is sometimes glycans can be buried in the protein structure, and then the enzyme can't get at the cleavage site, and so you don't release the glycan at all. And so there are methods to denature. Or you could, if you don't care about the protein at all, digest it potentially. So there are ways to do that. You could denature it with a chiotropic agent like SDS, and then you add a sequestering non-ionic detergent so that the SDS doesn't denature the PNGSF, and then it can act and release. But again, you lose some information on the protein. So it depends on the experiment. But if you're looking for release on glycans, there are chemical methods through beta elimination, and there are quite a few other endoglycosidases that you could look at, including EndoH, and there's a glycosidase from almond that doesn't care about fucose you can look at, and there are others as well. So.
Great, thanks. This question came in during your presentation. Could you suggest a best way to pull down all N-glycosylated protein in the cells?
Yeah, I saw that come in, and it's a great question, Nak. And if there isn't a very simple answer to that, but I'm going to give you like maybe a few ideas to go down. So the first thing, there's an old, an older method, sort of a traditional method for enriching oligosaccharides with like boronate columns. It's not going to be selective to N-linked, but you may want to look at that. We have like I think it's a aminophenyl boronic acid resin. So it's pretty non-specific, but you can capture glycoproteins with that. The method that's probably most used for if you need to be selective towards N-linked glycoproteins, that is probably lectins. There are antibody methods as well. Antibodies tend to have, for sugars, a lot of the antibodies derived against sugars tend to have really broad specificities, maybe low affinities. So you'd have to explore that. But there are lectins and lectin resins, and even mixed lectin resins to capture all the sorts of forms you might see in that tissue. So people will look at, they'll take cannabinoid A for high mannose, they'll take wheat germ agglutinin, they'll take jacqueline,
and a few others, and try to get at what they expect to see. These are lectins with specific specificities towards certain glycoforms. And if you have some idea of where it's coming from, is it human source, is it porcine, you know what you need to capture, and then you build your mixed resin based on that. So there's not a real simple universal single shot solution, but there are methods. And I recently put out like a lectin selection guide because it does get complicated. So if you care to, I can send that to you, and it kind of goes through some of the specificities of different lectins and what sort of products are out there to help with that.
Thanks. And there was another question from the same person. Do you provide a service to look at N-glycosylation profile in cells?
I personally do not. There is a group within our company called BioReliance, and they may do some of that sort of service, and maybe I can. I don't know how to direct you there off the cuff here, but I can definitely follow up.
Yeah, you can follow up with people. Great. This question also just came in. How important is it to have a known glycan structure, and where do untargeted glycomic studies fit into this?
Let's see. So how important it is, I guess maybe I would need a little clarification about in what context. So it's as important as it is to the researcher, depending on what they're doing. It can be very important in say pharmaceutical development, right? If you don't know the structure of your molecule in a therapeutic biologic, that's a problem, so that can be extremely important. So and then it can be important for physiological reasons, or maybe you don't care. Maybe you're just looking at protein and you don't care about the glycan. So it really depends on the researcher. So maybe a little clarity on what context you're asking about. But as for where the untargeted studies fit into this, I'd say Morton's talk was a great example. So they went in with this idea of using glycomics, really untargeted glycomics, to discover biomarkers or really like a glycomic thumbprint to track sepsis, and so that was untargeted. Eventually, the idea being there becomes a targeted approach that gets discovered. What that glycoprofile looks like that tells you sepsis gets discovered, and then it becomes targeted, and you start to look for those, and you can apply maybe more targeted mass spec approaches like triple quad stuff. You're looking for a specific signature. So, but the beginning, the discovery is very untargeted, the glycomics. I hope that answers what you're getting at. Feel free to clarify if not.
Yeah. In the meantime, I do have one more question for you. And that is, can you maybe give some pointers on how to go about preparing samples for a glycosylation analysis?
Yeah, that's a pretty broad question, and I appreciate it because it's I just maybe a few pointers that pitfalls that I can see I've seen over the years and things. One is know whether or not you're really interested in quantitation or qualitation. Are you trying to identify glycoforms, or are you trying to quantify some sort of distribution? And that know that before you start designing your experiment, and really consult with your mass spec, your analytical mass spec core facilities on how to quantify, because quantifying glycans can be quite difficult. They have variable ionizations depending on their attachments. They don't ionize very well. There are labels for that. So know what you want to do before going in, and maybe consult some analytical science experts to help with that. And then maybe the other thing I'd say is don't underestimate the power of glycosylation. So we can get caught up in the protein when if you're prepping a sample, you're making something, you need an enzyme activity, and it's not working. Something's not working in your experiment. Sometimes it's just keep glycosylation in the back of your head if you're not used to that. Sometimes we forget the importance, and it could totally. You saw like the example I gave on RNase. It could totally remove an activity or change how something interacts in a solution. So just keep that in mind through your experiments. It may be very important, and sometimes it's essential and critical.
And then one question that just came in: are there any basic differences between prokaryotic and eukaryotic organisms that people need to keep in mind?
I'd say for the vast majority of situations, you're not going to see end-link glycosylation in prokaryotic organisms. There are exceptions to that, so like anything in life sciences, but for the most part, and if you're in if you're working like in recombinant systems, the common hosts and things like E. coli do not have end-link glycosylation. They have very, very minimal sorts of post-translational modifications. But some of the like if you're into microbiome or environmental microbiology, you're going to see very complicated glycan structures like biofilms forming peptidoglycan interactions between the cells, almost like tissues. So it's not to say that it's simpler in a prokaryotic system. It's just different. So maybe before going into that, research that species that you're into, right, or that environment, and know what sorts of oligosaccharides to expect. That'll help you in your designs.
Thanks. I think that takes us to the end of our questions for today and the end of the webinar. So thanks again, Mark, and thanks to Morten as well for a very illuminating presentation and a great discussion that we just had, Mark. And thanks to our sponsor, Merck KGaA, Darmstadt, Germany. And finally, thanks to you, the audience, for taking the time to attend and listen in. So until next time, good luck in your research, and goodbye from all of us at Merck and Bite Size Bio.